Okay, maybe I'll get started now. Cool. So welcome everyone. Thanks for signing up for the tutorial. Today we will be doing some advanced single cell data analysis. In the past, we've had other tutorials for intermediate and sort of beginner levels. But today we'll sort of move forward and try to deal with the more complex areas that people are getting into and need more help with. Okay. So I hope that all of you have the Markdown downloaded and ready, and all of your installations are done, our RStudio and the Markdown. It'll be very easy for you to follow along during the tutorial. Okay, so a few points to note before we move forward is that this tutorial will be completely on single cell analysis with Monocle 3. And a good point to note is that Monocle 3 is a completely independent version of the tool. If any of you have been using the older versions like Monocle and Monocle 2, then you'll have to sort of redo your analysis because although some of the legacy functions have been ported over to Monocle 3, everything from literally making CDS onwards is different in the new Monocle from the older versions. And we're not really using the older versions anymore. This tool is, as you know, it's on GitHub and it's very often updated. So if you happen to use it quite frequently and notice any bugs in any of the functions downstream, then please submit detailed bug reports on the GitHub issues tab. And if you have specific questions that are not exactly bugs to report, but just general questions on how to do things, like what functions to use for doing specific kind of analysis, then there is also a Google group, and I've included the links to that in the Markdown so that you'll be able to access it easily. All right. So in the last two tutorials, we had one last year for an intermediate level, and one at the very beginning of this year that was a super beginner level, including installations. So today we will be covering differential analysis testing using Monocle 3. It has two methods, regression analysis and graph autocorrelation analysis, and we'll also talk about trajectory analysis. So today's rundown sort of is a recap of loading the data, a little bit of QC, pre-processing, and generating the reduced dimension coordinates of the data. And I'll be running through quite quickly through this section because we've done it a lot of times, and for the sake of time, we want to give more time for the differential expression and trajectory analysis. Then combining of CDS objects will point you towards the right functions and even batch correction. So we'll be using data, C. elegans data from Jun Kao's data set, a published data set from Jay Shendre's lab. But if you have different CDS objects that need to be combined and batch corrected, we'll point you to the right functions, and also show you how you can optimize your clustering if you're not super happy with it. And then we'll touch upon two methods to do differential expression testing, and also on how to subset your CDS and learn the graph and plot your trajectory and sort of check the changes in gene expression along trajectory that you've plotted or learned. So these three are this is the general workflow of the Monocle 3. If you've gone on the website, you would have seen this. These links are for the Monocle 3 website, the GitHub repository, and the Google group for questions. So please feel free to post anything that you may need help with. The team is generally on it most of the times when they're not busy with other things. Okay, so within sort of the larger areas that we're going to cover, some of the basic questions that we have when we do any kind of data analysis is first, how many good cells you have, which is determined by basically three factors. One is that you don't have very low UMI. You have filtered out very low UMI, then you don't have doublets in your data, and the third sort of parameter is that you make sure that you don't have mitochondrial read contamination. So in this section, which we are going to sort of run through, you can plot density plot for UMIs. You can filter for UMI counts and check the mitochondrial distribution as well. If you're working with nuclei, you shouldn't have that big of a problem with mitochondrial read distribution, but we generally suggest a threshold of 10% for mitochondrial reads. So keep all the data that has mitochondrial reads that are less than 10%. So next very common question is how many types of cells I have. So you do dimensionality reduction, or first pre-processing, which includes normalization, then dimensionality reduction and clustering. And one of the things that happens after you cluster is you're not really happy with the clustering. And if you have data from different runs or just very diverse samples in a massive run like NovaSeq, you would want to correct for batch effects. So for this purpose, you use sort of optimization techniques to make your clustering better. And there is a special function called align CDS function that I'll talk about when we start the markdown that helps you correct for batch effects in your data. Then the next question is like, what makes these type of cells different? So if there are two populations of cells that are very different in their gene expression, what sort of test should you use for differential expression? And if you have a time series of data, then what sort of test you should use? Because there are things to consider when you have a sort of developmental or time series data where you can see a clear path that the data is traveling along. But when you have conditions and controls, then there's a different test for it. So we'll sort of talk about that in detail. And then you also, sometimes if you have time series data, then your question is, does my data express different genes in different functional states? So for that, you would want to do trajectory analysis, which we'll also talk about. And depending on the biological question that you're trying to answer, you may have many more questions that you can try to find out from your data. Okay, so about questions and feedback, this is our first tutorial via Zoom. So bear with me if I am slow or get stuck in places. We've already had one glitch. We messed up and we forgot to have a chat box here. So you're gonna have to raise your hands if you have questions during the session. I'll be sending out a survey after the tutorial, so please feel free to give your recommendations and suggestions to improve the tutorial. We're constantly looking for good ideas. If some of you have attended or held or hosted some of these tutorials, then we're we're happy to learn how to make these sessions better. And yeah, the third point doesn't apply anymore. So just to check, is everyone able to raise their hand at least? Okay, perfect. Okay, I see it. Awesome. Great. So when we are moving through the tutorial, if you have questions that are related to that section, raise your hand so that we can address the questions. If you feel that you have questions that are not really in the scope of today's tutorial, then what you can do is you can email me. You can just reply to the email that I sent yesterday and ask questions. And with our feedback survey, we can sort of send you a transcript with all your questions answered. I also host office hours. So if you want to meet one-on-one, we can set that up as well. And I can send you a link to sign up for the office hours.
Ashwarya, it's Kim.
Yeah.
What about, one possibility for questions is if, I don't know if Kaylin's able to track them, if people could email Kaylin possibly, and then she could snap them in a chat box to you, that might work a little bit more effectively to see if people have the same questions and things.
Yeah, that works too. Yeah. How about we say if you have a very detailed question, you can email Kaylin. Kaylin, can you give your email ID?
Yeah, my email is cspur@UW, and I'll reply all to the thing that you sent out potentially.
Yeah, yesterday's email. Yeah.
So you'll all have my email, or you can if you have my Slack. You can Slack me. Either way it works.
Yeah, that works. If you have a super quick question, just raise your hand. It's fine. Cool. So I'll switch over to the to my RStudio now, and feel free to just work along the tutorial with me. If you feel that you're not quite ready and you need help on the beginning parts, then you can just watch, and then at the end of the session we'll have a couple of breakout rooms for those that need help with the sort of the beginning parts that have not attended the previous tutorials and are doing this for the first time. And we'll have this session or this room as well to just answer all the questions that you have. Hopefully we have time. Okay, so I'm gonna stop sharing. Okay, can you see the RStudio? Okay, great. So we'll stop sort of halfway, and I'll get a count on how many people have questions on that section, and then we can move on to the second half of the tutorial that might make the questions part easier. And Kaylin would have collected questions by then if there are any. Okay. So the structure of today's tutorial is sort of three main parts of the tutorial. Is the main content, which I'll be talking about as I run through the tutorial, on basically about what each section does, what the function does, and why you're doing that step in your entire flow of analysis. Second is the bonus questions. These are questions at the end of each main section of the tutorial that you can try to work through during the tutorial, and we'll give you a little time after each section to work through that. And challenge questions are those that are quite difficult, difficult as in not impossible, but you just need more time for it, and you don't have all the information that you need to sort of answer it like in the 2 hours that we have today. So you are free to try this when you go back and come back with questions if you have any. Okay, so this is sort of the basic things that I had in my slides as well, the links to all the helpful resources related to Monocle 3 that you may require if you are using it quite frequently. All right. So I'm going to run through the first section really quickly, and I'm going to tell you what I'm doing, but not going to talk about it in detail. So to begin with, I'm just loading Monocle 3, ggplot2, and dplyr libraries. I already have them installed, so I'm just loading them. Next is, since I'm using an already published dataset, the cow dataset, it's already available, and I'm just downloading the gene metadata, the cell metadata, and the expression matrix, and making my CDS object. And if you have your own data right now, you're free to work on that using the same markdown. The only thing is the column data or the columns in your call data may vary from this. So you'll have to change a few things in the lines of code to make it work. All right. Okay. Next section is if you already have a CDS in the RDS format, you can just read it in using the read RDS function. If you have multiple CDSs, you can use the combine CDS function to make a list of all the CDSs and then combine them into one mega CDS and then work through and do your analysis on that one CDS. Then next we'll check out the accessor functions. So this is the call data, the row data, and the counts. So the call data gives you all the columns that you have in your data, and the row data gives you the gene information, that's the gene ID and the gene short name. So as I said, good cells are high UMI, low mitochondrial reads, and singlets. So what we're going to do first is make a column called NUMI in your call data so that you can filter for UMI, low UMIs, and extremely high UMIs that aren't real. So in this step, I'm filtering for all UMI, filtering out all the nuclei that have UMI less than 500. And there is no set standard for what threshold you should use. It depends on your data completely. So you have to take a call, try different thresholds and see what works best for your data. This is just a density plot of the UMI distribution in your data. Okay, so one challenge question in this section is how do you find the distribution of the mitochondrial genome or reads in your data? And for this, what you have to do first is you should know what mitochondrial genes are there in your data and then sort of get that out and then count how many mitochondrial reads you have, which takes some time. So you can do it when you go back. Okay, next step is pre-processing the CDS. So the pre-processing step basically does just the normalization based on log and size factor. And feel free to stop me if you feel that something's not clear or you need more explanation of a certain part. So plot PC variance, what it does is it basically plots the the PCA components in your data. So it shows you at what number of dimensions will all of your variability, or will all the variability in your data be explained? So in this case, it's at 50. So basically, if you have number of dimensions 50 and above, all of your variability is going to be explained. But if you have a variable, or if you have a number, if you select number of dimensions as 20, there'll still be some variability that won't be accounted for. All right, next, I'm going to reduce the dimensions of the data. And when you use monocle 3, the default number of dimensions is 50, and you can change that. So for very complex experiments, you can increase it. And you can always plot this PC variance plot to check if you're accounting for all of the variability. All right. So I did, I ran a function called detect genes. So what that does is it adds a column called num genes expressed in your column data, and number of cells expressed in your row data. Next, what we'll do is we'll plot the cells. And if you look on the right, it's all grayed out because we haven't clustered it yet. So next step is to cluster the data. All right, so one of the main questions that we get is when people go through the Monocle 3 website, there is something called partitions and clusters. So the question is, what's the difference between partitions and clusters? So partitions are basically super clusters. So if you're doing a very exploratory kind of analysis and you just want to see how the gene expression changes happen across your data, you could use partitions. It also makes the analysis faster because it groups the data into bigger chunks. But it's possible that in your data you could have 10 partitions, and when you want to look at the clusters, each partition could have 4 different clusters. So you could have 40 clusters, but just 10 partitions. So let's see, in this data, I want to see how many partitions I have. So it says that I have 27 partitions. And how many clusters do I have? So I have 33 clusters. That's what I mean. So a partition can have clusters that are closely related to one another. Now let's plot after clustering the cells. And now you can see that it's colored by the different clusters, the 33 different clusters. And you can choose the color cells by option to color by whatever you like. Any column in your call data could be used to color your data. So it could be partitions, it could be clusters, it could be the number of UMI. It could also be like if you have a column that says this nuclei is a doublet or a singlet, then it could also be that, just to see how many doublets and singlets you have in your data. Okay. Next is if you want to plot the expression of specific genes, what you do is you combine or make a list of the few genes that you want to look at and then plot them. It's taking time. Yeah, and it tells you in what cluster that gene is most expressed. So, all right. So the question here is, you did your clustering and you sort of don't like the way it is clustered. Then what can you do? So how can you change it, or how can you optimize it? So there are several parameters that are available in the cluster cells function. So all you have to do is do !cluster cells. That is the helper function for cluster cells, and it will give you a description of what that function does and the different parameters that you can have in that function and what each parameter does. So in this case, for instance, what you can do is for optimizing your clustering, there are two things that you can change. The resolution, which is the parameter that controls the resolution of the clustering and how nice it looks when you cluster, and this integer k, which determines the number of nearest neighbors that are used when you're making your clusters. So one thing to note here is the k and the resolution are inversely proportional. So the higher your k, the lower your resolution is going to be. All right. Yeah, so what I'm going to do here is I'm just going to use some resolution and cluster the cells again, and then replot the cells. And I'm running through this section, but what you can do is you can download both the plots and see the differences, the previous plot and the one with the resolution parameter as well. So one of the bonus questions of this section is, can you try plotting other genes of importance to you? So this data is a worm data set. So I have worm genome genes in here. But if you are working with your own data, you can do it right now and see if it works and if you're able to plot. If you have if the list of your genes, that specific genes that you want to plot, is too long, your plot is going to be sort of squished together. So you would have to use ggplot2 on the side and sort of change the dimensions of the plot and make all the genes fit. All right. So the questions 1 and 2 were sort of all the beginning parts that we have already done, so I didn't want to spend too much time on it. The third question is, how do you correct for batch effects for data coming from different sequencing runs? So the align CDS function in Monocle 3 helps you correct for batch effects. So this function uses the bachelor package and helps you to align data. You can choose the pre-processing method. PCA is generally for RNA-seq analysis, and LSI is generally more robust for ATAC-seq analysis. Then you can change the model formula string, so that helps you decide what you want to correct for. So if you have different plates, for instance, and you want to correct for batch effects across plates, then your residual model formula string would be plate. So you could add those parameters and remove all the unwanted batch effects that are present in your data. Okay. So what I'm going to do right now is this data that I'm working with has I think around 12 plates of data. So I'm going to use plate as my alignment group. And since it's a large data set, I'm going to use 100 number of dimensions and align my CDS. And when you're running things in the console, this is a stupid thing, but every time you see red, it doesn't mean that you have an error. It can also be a helpful message sometimes. So one important thing to note is, after you align your CDS to correct for batch effects, you want to reduce dimensions again and then plot your cells, because the alignment or the batch correction won't be accounted for if you're not reducing dimensions again. And there are a few things, these small details that are not mentioned, and then you happen to just go down a rabbit hole and it's really hard to come back. Okay, so what I'm doing now is I'm plotting the cells and I'm coloring by plate, and it should look good because I just corrected for my batch effects and my alignment group was plate. Okay, so if you see this plot, you can see that all cells fall evenly across all the different clusters when you batch correct by plate. And you can use different variables in your column data to check that your batch correction actually worked. Yeah, so if the bonus question is, if you want to batch correct based on any other variable in your data, you can always try it with your own data and see if that helps at all. Okay, so next we have 2 or 3 main questions that are related to differential expression analysis and trajectory analysis. So before we proceed, are there any like fundamental questions about the first 3 parts that I covered? Was there anything unclear, or can I proceed? So if I can proceed, raise your hands. And Kaylin, do we have any questions? Okay. Perfect. Okay, then I'll move ahead. Okay, so the next question, a very main question, sort of the basis for doing single cell sequencing at all, is what makes the type of cells different. So for this, we will run through differential expression testing. So the sort of different questions that come up here are these populations of cells different in their expression of specific genes, or are all the genes drastically changing? Then what are all the genes that are differentially expressed? And can you correct by, or can you add certain variables while you do differential testing to see if you have different conditions, and if because of those conditions your gene expression is changing across your, say, cases and controls. And then there is something called modules of expression, which is nothing but gene co-regulation. So there are, as we know, there are a bunch of genes that are sort of regulated in similar ways. So you can see if their gene expression patterns or are changing in tandem. So, all right. So since I'm using the worm data set, I have some very clear markers for body wall muscle markers and germline markers. So I'm going to use that to plot my cells first. I think I already showed this step previously. Okay. Next, what I'm going to do is, so there are two methods for differential expression analysis. One is regression analysis that uses the fit models function, and another is the graph autocorrelation test that uses the graph test function. So first we'll go over the regression analysis. So basically we use regression analysis when there is an individual variable of dimensionality reduction, such as cases versus controls, or healthy versus these many time points after treatment. And for this sort of A and B sort of analysis, pseudo time or trajectory analysis is not the best way to go. So this is when we use the regression analysis and use the fit models function to do the differential expression testing. So what I'm going to do here is I'm going to first subset my CDS, and for subsetting, so what I want to do is I have 33 clusters now in my data, like you can see in the UMAP. What I'm going to do is I want to choose any two clusters and see the genes that are differentially expressed between those clusters. So for that, I'm going to use the choose cells function. And when I do that, what happens is a pop-up, a shiny interactive window pops up. So I'm going to have to switch between that. So there's going to be some lag probably. All right, so I'm going to stop sharing my RStudio and go to the shiny part of it. All right. Can you see? Okay, so you have your UMAP displayed here. So the choose cells function helps you choose n number of clusters and then calculate the differential expression of genes across those chosen clusters. So what I'm going to do for the sake of time and quickness is I'm going to choose two small clusters. So if you just drag around the cluster and say choose until it becomes black, and then I'm going to choose this one. Say choose. So both of these are black now. So these two clusters have been chosen, and then say done. And as soon as you say done, the pop-up window disappears. That's why the sharing has stopped. Okay, so I'm going to go back to my RStudio. Okay, so two clusters have been chosen now. So the next step is to use the fit models function to calculate the differential expression of genes across these clusters. Model formula string is the variable or the variable that you can use in your regression analysis to check for differential expression across different parameters in your column data. So one thing to note is the model formula string should always be in your call data. It can either be clusters or anything in your call data. So if it's not present in your call data, it's not going to work. And again, I'm using only the first 1,000 genes here because even after choosing the smallest clusters, it's still a large data set. So I want to not have it hang here. So I'm going to use the first 1,000 genes in my subsetted CDS and do the fit models analysis. It won't be the best of the results, but it'll give you an idea of what happens. So when you use your entire CDS, it's going to take a while. It's going to be exponentially longer. All right. Okay, so that was quick since it was just the first 1,000 genes. Next step is to get a coefficient table from your regression analysis, just to better help you look at what the output of the regression analysis or the differential expression analysis is. So we store that in a variable called fitcoefs. And then if you do a head of fitcoefs, then you can see that it's a table of 6 rows by 13 columns. And all of the 13 columns are not seen here, but you can see what the different columns are. So, and you can see that all of the genes are like all A's because I just literally took the first 1,000 and it's alphabetical. So what this, all right, so what I'm going to do here Okay, why don't I move ahead and then explain what the table means? So when I did fit models here, I used cluster as my model formula string. But what if I want to look for differential expression with more number of variables included in my fit models function? So the next step is to add another model formula string. So I'm again going to add. So I batch corrected by plate earlier. That means that I have a plate column in my call data. And I'm going to add cluster plus plate. So that means that, so a very simple example of these dual or two or more variables in your regression is that suppose you have age and cancer and no cancer as your two variables. So what it means is if you keep age constant, then what are the gene expression changes between cancer versus no cancer? Or vice versa, if you keep the cancer status constant, then how does the expression change across age groups? Something like that. I hope that was clear. So you can add however many extra variables you want in your analysis, depending on what you're trying to answer, what question you're trying to answer. So I'm going to do the fit models test again, but with an extra variable called plate. And then we'll see what output we get. All right. So next is again getting the coefficient table from your fit models output. And then if we look at fit coefs now, do you see that the number of genes have increased because I added extra variables, and now more of your gene expression changes could be explained. So it totally depends on what you want to add as your variables. So this is a large table. If you can see that it has 13,000 lines and 13 or rows and 13 columns. So how do you interpret this table? So there are 13 columns in the table, and some of the important columns that you might want to access are the term, which is here, estimate, normalized estimate, p-value, and q-value. So the term is basically you're saying what you want to find differential expression against. So it's either plate 1, plate 2, or cluster 12, cluster 6. It could be anything, anything that is in your call data. Then the estimate is, like I said, keeping one variable constant. What are the gene expression changes across another variable? And the estimate is a number. It gives it's an integer, so it is either negative or positive, and it will tell you whether a gene's expression is decreasing if it's negative and increasing if it's positive. And the normalized estimate is what sort of gives you the magnitude of change and makes it easy to compare across genes among those clusters that you have chosen. So if you want the magnitude of change, you would want to access the normalized estimate column. And there's always p-value and q-value to tell you if the test is significant or not. So one thing that you could do is you could take this table, export it into a CSV or an Excel file, and then use any plotting functions that you usually use to plot the different values for the different genes that are in the table. So it makes it very easy to sort of move away from the monocle tree workflow and do some plotting on the side if you want to. So next, what we're going to do is we're going to find significant genes, say in cluster 12. So I'm going to take my fit coefs table and I'm going to filter for the term. So the term part comes again, the cow cluster 12, which is how it's named in my column data. And then I'm also going to filter and control for the false discovery rate by setting a threshold of 0.05 for the Q value. And the only thing that I want to output from my large table is the gene short name, the term component, the Q value, and the estimate. You can have the normalized estimate and several other things. You can have all 13 columns output, no problem. Great. So now that I have control for Q value, you can see that there are 10 genes or 10 rows and then 4 columns that I selected that I want to output. So, okay. So basically here, my model formula string were the two things, cluster and plate. But what you can have different things like time or developmental stage or time after like response to stimuli or drug treatment, anything. So as long as it's a legitimate value to use in the function, it's going to work. Next is, so a very basic question is when we do bulk RNA-seq, we do a lot of box plot and violent plot and heat mapping. And that's one thing that is a common question of how to actually extract the necessary information from this huge data set and plot a simple plot. So one of the things that you can do is you can plot the violin plots for gene expression across different clusters. Again, anything in your call data. So I'm going to use my CDS subset, which is just the 2 clusters that I selected, and I'm going to try to plot violin plots of gene expression. So do you see the error that I get? Since my CDS subset have way more genes than 100, it's not possible in that little space to plot so many violin plots. That's why I'm getting the error. So what I'm going to do is I'm going to select some genes of interest to me and make a list of those genes. So I'm making a list of ciliated genes here. And then what I'm going to do is I'm going to make a new ciliated genes CDS that has all the genes or all of those 6 genes. So it's a very tiny CDS only with those genes so that the analysis is quick. And this helps a lot when you already know what your genes of interest are and you want to quickly look at it. So I make a new CDS here. So if you see, if you just do CDS ciliated, then you can see that the dimensions of the CDS are 6 and then 22,500. So it says that you have just 6 genes that you've chosen. Okay. And then you can plot the violin plots for this CDS and then it will plot. So what I'm saying is I want to plot violin plot using this new ciliated CDS, the tiny one. And then I want to group the cells by the cluster. So let's see how that looks. Okay, it still looks very messy because as you know, we have 33 clusters in our data. So it's going to be all squished together. So if you still want this plot, you're going to have to sort of change the X and Y dimensions of the entire plot to make it fit. So what I could do is next I'm going to group the cells by the tissue type, which is another column in my call data. So that may help make it cleaner. Okay, so the tissue type is displayed along the x-axis and the expression along the y-axis for the 6 genes that you have in your list. So this is how you plot violin plots. You can plot box plots. You can also use the p heat map function or the heat map 2 function in R to make heat maps with the same values. All right. So one of the challenge questions in this is literally the first is totally dependent on yours. Like what else do you want to find out in your data? Does this sort of analysis help you answer your questions, or do you have different questions? And then based on the variables in your data, what variables would you like to use to test your data, which is nothing but the additional variables in the model formula string. So this is the first way of doing differential expression analysis, which is the regression analysis. The second method is graph autocorrelation analysis. And again, before I move to this part, are there any major questions, or does any section need more explanation? Is it all okay to move ahead? Perfect. And just feel free to answer, like, try to work through these bonus and challenge questions. It'll help a lot because you'll get a basic idea of what are all the first steps you should do to analyze your data before you go into something extremely complex. So yeah, try to answer. And if you have questions, feel free to email me. Okay, so the second method is graph autocorrelation analysis. So this type of differential expression analysis is used when you have a nice structure in your UMAP, a very sort of interesting structure in your UMAP. Then it is also used, it's basically used, or always used in conjunction with trajectory analysis. This is because if you want to see if there is a process that is happening across pseudo time or across a given trajectory that you have learned. And then in this sort of analysis, you're basically asking if the expression of nearby cells in your UMAP gives you some information about your cells. So if you say that this is my starting point, then there are some group of cells that are nearby, and you want to test their differential expression to see, or test their expression to see if they are highly variable or very similar to your cells, which informs you whether those group of cells are very similar to your starting point, or they're gradually changing over time. Great. All right, so we already have a list of ciliated genes. We've already made this ciliated genes CDS. So what I'm going to do is just use that ciliated CDS and okay. So this new CDS, that is the ciliated genes CDS, is not yet clustered. So we clustered the cells again. Okay, and then plot again by partition. Okay, so for this analysis, like you used fit models function in the regression analysis, the main function used for the graph autocorrelation is the graph test. So I'm running the graph test function on my tiny CDS using the neighbor graph method as KNN, and you could choose other methods. I'm just saying that I want this much memory, 8 cores of memory, and then I'm storing it in a variable called PR graph test result, or principal graph test result. And if you go to the Monocle 3 website, it will tell you when you should use different algorithms, or different methods for nearest neighbor. It's either KNN, or for other parts of the analysis, do you have to use negative binomial or quasi-Poisson? So it's there are different instances when you have to use, or when certain methods will be more beneficial over the others, and that information is given quite clearly on the website. So next what I'll do is I'm going to again control for false discovery rate by giving a threshold for my Q value of 0.05. All right. Okay. So my test is basically done. So again, what you can do is you can write this result to a CSV file and again, look at the columns and then use ggplot or any other plotting tool that you're comfortable with and plot the results. So let's, so basically you just do write.csv and then save that data frame into an Excel file. So what this output will, so let's see, head. Okay, so if you see this PR graph test output has several different columns like gene ID, gene symbol, p-value, q-value, and a number, a count of the number of cells that are expressed above a minimum threshold, and then a statistic called a Moran statistic. So basically, if you want to sort of rank the genes based on how similarly or not similarly they are expressed, then you can access the Moran's 1 column of this output, and which will help you rank the genes and look at their expression side by side. So sort of the take home from this section is that you used fit models function when you investigate genes that are differentially expressed across clusters, and it's a regression analysis. And then graph test is an alternative way that is always used in clubbed with trajectory analysis for finding genes that vary across groups of cells, but in a UMAP or t-SNE space. Yeah, so again, like I said, the bonus question is that you can rank the genes by how similar or different they are in their expression values by accessing the test statistic in your output and plotting the expression. And you can also filter for P and Q values because that'll be in your output. Great. So what does this Moran's 1 test statistic actually mean? So you'll have different values from -1 to +1. So 0 means that there is no significant effects, and +1 indicates that there is a positive autocorrelation, and which means that the nearby cells have very similar gene expression values to your starting group of cells. So if you're moving along a trajectory and you said that this is my starting point, then if further along the trajectory, if the Moran's test value is +1, it's going to mean that those cells are very similar in expression to your beginning group of cells. And that value may change along the, as you move along the trajectory path. So it shows that a gene is expressed in that focal region in the 3D UMAP space or three-dimensional space. Yeah. So the challenge question in this part is for you to use all of this information and see if you can find genes that are co-regulated. So there is a special function for this called find gene modules. So it helps you find genes that are regulated in tandem, and then you can also do pathway analysis on those sets of genes to see what sort of pathways are up or down regulated, like GSEA, David, GO, basically. Yeah, so that sort of takes you towards the gene set enrichment analysis testing. And that again will take time, and we don't have a lot of time. So we're not going to cover that. But has everyone been able to follow along until here? And are there questions about specific steps? Because next part is the trajectory analysis part, after which is pretty much our last segment. So it'll take about half an hour more. So if you have questions, now is a good time, or if you need a break from my talking. Yeah, Kaylin, do we have questions? Okay. Okay, so one question is, have people actually done this, or is this completely new, or you've done parts of it and didn't quite know how to do other parts? It would be nice for us to gauge how much people have done in the background before actually coming to the tutorials. So for those who have tried, any input here for how much you'd already tried in the past?
Aishwarya, I don't know if this has happened to anybody else, but after I did the CDS subset and my 2 clusters, it totally kicked me off R during the tutorial. So I just had to reload. I had to start a new session and reload. But
so the pop-up worked, and then you chose the 2 clusters.
Trying to manipulate it, I think my R just got maybe or my computer got overwhelmed.
Yeah, one thing you might want to do is like check that you don't have other big processes running in the background, because for those small clusters, it should technically not give any, or it shouldn't make your computer mad. Cool. I hope everyone's been able to follow along, and please stop me. We have time, so we're not running out of time. So feel free to stop me. I think I got a couple of emails about questions, but maybe we can get to it after the next section is complete. Okay, so the next part is the trajectory analysis. So the basic steps to do the trajectory for constructing trajectories is very similar to how we do clustering, but there are a couple of other steps that need to go in before the trajectory is actually learned. So I'm going to make a new CDS that has only 1 cluster. Because there are 33 clusters in this data set, I don't want to have to wait around for it to learn trajectories for within each cluster. So I'm just going to pull out cluster number 6 and make a whole new CDS with just this cluster. So that's what I'm going to do now. And you don't have to do this when you run your own data, run it on the whole CDS. Just make sure that you have, if you're running it on the cluster, then you have enough memory. And if you're running it on the laptop, that your laptop is up for it. Okay. And as we know, once we subset a CDS, it needs to be normalized and have its dimensions reduced and clustered again. So let's do that. Okay, so now that I've subsetted the cluster 6 from my big data set and made a smaller CDS, I have clustered it. Now the next step is to learn the trajectory graph within this cluster. So the learn graph function in Monocle 3 will help you learn the trajectory within a cluster and sort of get the progression of cells in that cluster. So, oh, one other thing is, when do you see I replaced my subsetted CDS with the learn graph CDS. So if you want to keep your initial CDS intact, you can always put it in a new CDS, but that just makes you have like 10 different CDSs and then you forget what's what. So if you're not sure, or if it's not intuitive, your naming is not intuitive, then don't do it. Just keep replacing or writing over your old CDS, but yeah, otherwise you're just gonna have to do everything again. All right. So basically, this learn graph function fits a principal graph within each partition. So in this case, the whole of cluster 6 will be taken into account because it was a small subset of the whole of the data, and then it'll learn a principal graph within that cluster. And this part is much slower when it's your whole CDS. So bear that in mind. And then I'm going to plot the cells based on cell type. So I'm just going to color by the cell type. So if you see that it's just the cluster 6 and it's sort of try to learn the graph within that cluster. It is a small cluster, so it's not going to be the most perfectly learned graph because it's not a big data set. But it should give an idea of how it's going to look in your big data set. Next question is, are there any genes that are differentially expressed based on dynamics? So what you want to do is, you've learned your trajectory. Now you want to know that along this path, is the gene expression changing as you move along from the starting point to the end point? So for this, you're going to use the function called order cells that will help you learn if the expression is changing along the trajectory, which is nothing but a pseudo time analysis. So for this, if you don't mention what the beginning of your trajectory is as sort of a parameter in this step, then it's again going to pop up a interactive shiny page for me to select what part of the trajectory I want as my starting point. So I'm again going to have to switch. Let's do this. Can you see the root nodes shiny pop up? Okay. So this is basically the trajectory within that tiny cluster that I chose as my entire data set. So what I'm going to do is I'm going to choose this end and say that this is my starting. And say choose. And you see it becomes red, and then I say done. Okay, go back to RStudio. So now it knows what to consider as the beginning of my trajectory. And now it's going to take that as my root point and then sort of check the differentially expressed genes across the path. So what I'm going to do is I'm going to plot the cells, use the subsetted CDS, plot the cells in pseudo time, which I have learned by saying that my graph starts at that particular root node, and then see how it looks. So if you see this, it shows that there's sort of like lower expression in the beginning, and as you go, as you go higher, then the expression may be increasing. And this may not be super correct because I've just chosen one cluster, but you'd want to take your entire data set and check. But it's also possible that your data set is extremely complex and you just want to learn what the changes in expression along the path are for one branch of the trajectory. So you could take the clusters that are a part of that branch and then sort of check for how the expression changes along a particular branch of a trajectory. So it's possible that you have progenitor cells and then, through the stages of development, and you want to see if it's very different in the beginning versus if it goes into this lineage or lineage B or lineage C. And you can choose those different branches of the trajectory and calculate the gene expression changes. Okay, so the challenge question is if you can combine question 4, which is, or rather question 4 part B, which was the autocorrelation analysis, and question 5, which is the pseudo-time analysis, to see if to see how the gene expression is changing. So you are going to have to club these two parts, and because the graph autocorrelation test will actually give you the magnitude of changes for your gene expression, and the pseudo-time and trajectory part will actually tell you how the progression or how the branching occurs. So you want to merge these two components and then calculate your gene expression changes. You can plot them, you can download them as a as a CSV file and then plot them in a like violin or box plots or whatever you like, but you can also plot them in a UMAP. So this again takes a lot of time, clubbing the two, and because you need some output from graph autocorrelation and some output from the trajectory analysis, club them together and then get your final result. Great. So pseudo-time analysis is the main component of the trajectory part, and it's always done clubbed with the graph test, because just learning the graph is not going to tell you anything. You want to see the gene expression changes in the end. That's why we do RNA-seq. So that's one of the big, so you have all right now through this tutorial, you have all the functions for the graph test as well as the pseudo-time test. So when you go back on your data, you can merge the two steps and see if you can see significant and reliable changes in gene expression along your different branches if you have time series data. Also, one other thing to mention is when you clearly don't have progression data, it's not worth it to do trajectory analysis. It's just not going to give you, it's going to give you inconclusive results, which is going to confuse the analysis even more. Fit models works much better in that case, and it's quite robust. So, and you can always add these extra variables, which helps with the with accounting for the different variables in your data. So take a call based on what the data is. Great. Next, it's a very quick. Okay, before I move on, again, any questions in the trajectory analysis part? And we will have further tutorials based on your responses and your feedback. If you feel that, a section was helpful, but you would like to have much more detail, and you'd want to have like an hour and a half just on trajectory analysis, then we're ready to do that. Just give us a completely honest feedback on what you want to have us present for the next tutorials. But if there are no questions, can you raise your hand so that I can move?
I have a question.
Yeah.
Can you set the seed for when you're doing this trajectory analysis so that it always comes out exactly the same way that you would want to see it, or?
Right. Yeah. So that's a good point. So there's a parameter called, or rather a setting called set seed that you can set to like 1, 2, 3, 4, 5, or so it's a random number generator. So what happens is, like, when you're doing analysis, you're not going to do all of your analysis in one single day. So when you come back to it, and then people have had different, sort of slightly different or varying results every time they try to do an analysis. So what happens is, some steps in the analysis have random number generators, which are going to give you slightly different results when you do the analysis every single time. But if you set the seed to a particular value, then it's going to be reproducible every time you do the analysis. So you can just, like, literally Google set seed, and it'll give you all the information that you need. It's super simple. It's just one line that you add and run when you do the analysis, and it'll help you not have varying results, and you don't have to panic. Great. Okay. So this next section is a quick bonus section. It's very small, and it's like tiny questions that I've had during office hours that I'm addressing here. Many of you may already know it, but I'm just going to go over it. So one is, how do you select a specific cluster when you have 20 different clusters in your data? And then how do you take that cluster and then further divide it into subclusters? So it's super simple. We already did the first part in the differential expression and the trajectory analysis. But what I'm going to do is, I'm going to take my main CDS and just take out, or like choose cluster 1 and make a whole new CDS. So what I'm saying is, from my call data of the CDS, there is a column called cow cluster. So I'm going to choose the cluster 1 from that, and then take all the rows. So you know that in R, anything that's between a bracket, it's rows comma columns. So I'm saying that for the columns for cluster 1, take all the rows and make a new CDS. So just made a new CDS, and then I'm going to pre-process it, reduce dimension, and recluster. And this is a very small cluster, so it's going to give me error, and it's not going to look good. It's just going to be dots on the plot, because this is very small. But you will have much larger data sets where it might help. Okay, so do you see it's just like a starry night sky, because it was literally the smallest cluster that I just remade into a new CDS. So this is a simple, this literally one line of code is a way of sort of choosing a specific cluster. And if the cluster that you're choosing or you're subsetting out is big enough, then when you cluster that cluster again, then it will branch out into different clusters. Here it was very small, so you don't see anything on the plot. And then you can see, and when you recluster is when you see that it's not looking very great. So that's when the resolution and the K parameter for optimizing your clustering comes handy. And then how do I add a new, next question is, how do I add a new column in my call data? It's literally like how you add it in a data frame. So you just do a $sign with whatever cluster you want to add, and then you access the cluster slots in your cell data set and say that I want to take the cluster 1 and then add it as a column in my call data. As simple as that. All right. And next part is, we're not going to do anything, but this is a very, very important part when you're working with single cell data analysis. You want to absolutely ensure that you're working with good data and you don't have any doublets in your data. At the BBI, we use Scrublet in our pipeline to call all the predicted doublets and give it And when we return data to you, we add a column in your CDS or rather 2 columns in your CDS. One gives you a call, whether it's a singlet or a doublet. And then another column is the KNN score, which you can use as a threshold to sort of remove the doublets and only keep the good data in your data set. So we have looked at other doublet removal tools, and there's no good or bad here, but it's just for the sake of it. The performance, we realized that the performance is sort of similar across 3 or 4 tools, but the speed varies substantially. So Scrublet is a good combination of both, so we decided to go with that. It has great documentation. What it does is it takes your UMI counts matrix, and that has cells as rows and genes as columns. Then it calculates a doublet score for each of the cells or nuclei. And then it forms like simulated doublets from your data and uses the K nearest neighbor classifier to calculate a doublet score for you, which is anything between 0 and 1. And then you can use this KNN score as a threshold to remove the bad data or the doublets and only keep the singlets. So I've also given you the link to the GitHub repository for Scrublet. Do use it. It's very, it's well documented, and I think it's very easy to use, but if you have trouble installing and using it, feel free to reach out and I'll be happy to help. And so for biologists in particular, when they're working on the data, they are very amazed when it can all be aggregated together. All your analysis can be aggregated into a single document. So this is done by knitter or knitr, however you like to call it. So you can literally, all of this analysis that we've done from the beginning to end, you can knit it in the form of, or basically what it says is, it gives you the code as well as the result, chunk by chunk, and knits it all together into either a PDF, a Word document, or an HTML. So you can use that, and the only thing you have to do is say knit. So you can either do it as HTML, PDF, or Word. Super simple. It takes like a couple of minutes and you have a nice handy report to show. So yeah, this is the last part. So thank you all for attending, and we will have a tutorial every 3 months at least. And the next few might be on Zoom, but I'll send out the survey in the next 2 or 3 days, and or before the week is up. And please give us your feedback. It's really useful. And if you have the questions that people have sent, we can get to it now because we still have half an hour. And if you have more questions, email them to us, and then we'll give you the answers. So does anyone have questions at this point? And do we need breakout rooms?
So Aishwarya, here's a question. Can you clarify when you would use regression analysis and graph autocorrelation analysis?
Yeah. So regression analysis is basically used when there is like an individual variable in your dimensionality reduction. So very simple explanation is, if your data has cases and controls, so you're comparing A versus B, then regression or regression analysis with the fit models function is the most helpful. It can also be a healthy patient versus cancer patient, or control versus drug treatment. So when you have basically A versus B, that's when you would want to use regression analysis. Also, it's not just A versus B. So if you have healthy versus drug treated, and you have males and females, and then you have age groups of those, that can still be used because, as we covered, the model formula string helps you add these extra terms to account for when you're calculating when the model is running and when it's calculating the differential expression. So you can add age, you can add the sex, then you can add the treatment. As long as the information is there in your column data, it can be used to calculate differential expression. And the instance when you use graph autocorrelation analysis is when you are doing trajectory analysis. And when do you do trajectory analysis is when you have time series data or developmental data or data where there is a clear response to stimuli over these many hours. That's when you want that's when you want to learn a graph and know the changes in gene expression along the path of the trajectory and its branches as well. That's when you use graph autocorrelation, because it's in the UMAP space and it's not like a one-on-one sort of analysis. Any other question?
So we got another question about just like setting the seed, or just about how every time you run the trajectory analysis, it looks slightly different. So for example, when I ran it based off of your code, even though we were doing the same exact thing, it looked slightly different. So maybe can you talk a little bit more about why you see that slight variability?
So I'm going to punt this question to Brent, because he's had conversations with people that have had to do this. Brent, can you shed some light on setting the seed?
Certainly. So there are functions that use a random number generator, and the random number generator just creates a sequence of pseudo-random numbers, actually. However, the seed can be used to set the random number generator to a specific starting state. So this function set.seed sets the R's random number generator to a specific state given the seed number. There are also in certain functions a parameter for setting the random number generator seed. And so if you're careful about setting the seed, you should get consistent results from run to run when you perform the same analysis repeat the same analysis.
And Brent, just to clarify, you could literally set the seed to any number that you want, right? Like you'd always just say like set seed 1 is 1, or set seed equals 1, 2, 3, 4. Or...
Yes, it can be any positive integer. 0, I believe, will not work, and negative numbers won't work.
Aishwarya, I sent you a couple questions by Slack too. Just one.
Okay, let me check.
Bigger.
Okay, are the old tutorials posted somewhere is the first question. We are in the process of doing that. So I'm working on a side project where we have, as you all know, we have a BBI website, and I'm in the process of adding a separate page on that website that will completely cover like tutorials and resources that we provide. And I'll make sure that I upload all of the slides and markdowns for all the previous tutorials that we've had, and we'll mention what level analysis it is, so that it's easy for those beginning the analysis as well as for advanced users. But we're in the process of doing that, and that'll be coming soon, before the end of this year. But if you reach out to me, I am happy to share the previous markdowns, at least in the time being. Next question is, when I run gene module analysis using Okay, so the question is, when I run gene module analysis using neighbor graph, principal graph on my own combined CDS file, after making a pseudotime trajectory graph, I get uniformly high or low expression with little variability across UMAP space. Other members of my lab get similar results. Adjusting Q value or resolution does not help with this issue. Do you have any suggestions on how to approach this? So my very basic question would be, is it a time series data or a data in which you clearly know that you're going to have a progression or variability seen at every step of the way or the path? So based on that, it's possible. So one of the reasons is if you have data that is clearly not for trajectory analysis, you can use the fit models function and try if these changes are actually seen better when you use the fit models function rather than the autocorrelation. But if you have a clear trajectory analysis, then why don't, one of the things that you could try is when you plot the trajectory, you can subset the trajectories and check for gene expression in parts of the trajectories instead of the whole thing at once, so that you see more subtle results, I could say. Does that make sense? Yeah, because when you plot the trajectory, you can have 4 different branches. So one of the ways of looking at it, if you clearly, if this kind of analysis is clearly for your data, right for your data, then you can subset out the different branches of the trajectory and then try to see if the variability is seen more clearly than when you look at it as a big chunk. And we can talk more offline if you want to talk more in detail. Those were the 2 questions on Slack. Are there any other questions?
So that looks like it for now. I know this is really fast, so if you want to do breakout rooms to try and go back, that would be.
Yeah,
great.
we still have 20 minutes remaining. So if any of you need help with the sort of the beginning sections or the later parts, then we're happy to make breakout rooms. Let us know, I guess.
Hey,
Okay.
Caitlin, I guess I had a question for earlier on about what if we don't like how our cluster looks and like how we can approach redoing that. So we did ours on, I believe, a population of cells that might not have the highest like hierarchy. So the hierarchy might be quite shallow. So when I went and I played around with our first run with our data and went to cluster, it just made one cluster with all of our data because I think it's not very deep. But is there a way to, I guess, change the parameters to make it more fine-tuned or a high resolution to maybe piece out, like, even though it's not the deepest population of cells, to see within that, is there any like subtle changes amongst the different cell types?
First question is, how deep is or rather, what's the number of your recovered nuclei or cells?
From the data set I was using, I think it was maybe 30,000.
Okay, so is it a combined CDS or one single CDS?
One single one.
Okay, and you've done all the filtering and the basic steps in the beginning?
The basic steps, at least, like processing and reducing dimensions and all that. Yeah.
Okay, so one thing before you proceed would be to check for doublets. Just make sure that you're not working with any doublets, and Another thing would be to, so your question is, how do you improve the resolution and make it cluster better?
Yeah, if possible.
Yeah, so that's sort of a trial and error method. So one thing is you can use the K and the resolution parameter and sort of try different combinations of it to see if your clustering gets better. Another way to look at it is test if your optimization is working, is just subset out a part of your data and see if things very clearly change when you change the resolution parameter. If it doesn't, that means that the number of recovered nuclei is high, but you just don't have very or the depth of the data is low. Yeah.
Thank you.
Any other questions? I see one raised hand, but I don't know if it's for a question or maybe not. Okay, then I guess we'll wrap up for today. Thanks for attending, and please give us feedback, and I hope that this was helpful for you. And feel free to reach out by email, or we can set up office hours and go over any question that you may have in more detail. And I generally have office hours for 1 hour slot office hours every week, at least 4 days a week. So I'm happy to talk to you in detail separately. So yeah, thanks, and stay tuned for further tutorials. We have all of you on our email list, so you'll get the emails.
Thank you.
Thank you.